Hello, I'm going to do a short video about the lab, the genetics lab that you're going to be doing, I think the week after Easter. And so we're going to go through it because I want to point out just a few things and make sure you know of a few deadlines so that you're not confused. So I have this calendar on my phone where I wrote down all the dates. And so your genetics lab, it should open up on the 14th, so the day after Easter Monday, and it'll close on Tuesday, April 21st. I feel like I said December somewhere in there, and if I did not mean December, just to be clear. I just discovered today is actually April and no longer March, because I've been writing March on everything. Okay, so you're going to have your handout. It's all about genetics. Genetics is a really important part of the TEKS. In fact, there's like one whole competency on it, and usually only one or two questions for y'all to even answer, which is wild to me. So I want to point out the pennant square. Okay, so this is kind of like the weird math they're doing nowadays, where you write the numbers across and down, and you write numbers in between. You add everything up, but the capital letters are dominant, so meaning that they're more likely to show up. They're like dark hair is dominant over blonde hair. So if you have the RR allele for brown hair, just say that's what it is, you're going to be born with brown hair. So this is your genotype. It's what your gene says that your hair color will be, and the actual color that you see, brown, blonde, red, that's your phenotype. It's the physical expression of your genes. Lowercase are considered to be recessive, so they can be masked. If they have a dominant allele in front of them, then they are going to be masked and they're not going to show up. So recessive genes are not going to show up. This is the last thing I'm doing. It's day 32 of quarantine. We're fine. So then say that this is your parent, RR, red plant, and the other parent is, that doesn't match up well. But your other parent's genes are capital R, lowercase r, which because it has the capital R, it's going to show up red. So it'd be two red plants hybridizing. And then this is the likely color of their offspring. So you combine R and R, R and R, R and lowercase r, and then R and lowercase r. So 50% of the offspring are going to have the genotype capital R, capital R. The other 50% are going to have the genotype capital R, lowercase r. But their phenotype, they're all going to be 100% red. There are no lowercase r's, which would be the recessive color, which would be a phenotype of white. Okay, so that's how the percentages work. So, and there's lots of introductory material. You do need to read that. I would recommend it on this lab. All right, so the first thing you're going to do is a procedure that's based on coins, flipping coins. It doesn't matter what kind of coins you have, but you're going to need at least, I guess, two of the same coin, and you're going to need two of them. So if you have like 2 nickels, 2 quarters, 2 pennies, 2 dimes, it doesn't matter what kind of coin you have as long as you have two, and I would say they need to be the same type. So the first thing you're going to do is these are our two parental genotypes, and it wants you to fill in the Punnett square. So you're going to bring down the H and the H, the H and the H, and you're going to write them in. And you're going to say, it says determine the expected genotypes and phenotypes for the Punnett square. And so this is percentage. You know, it's going to be 25% capital H, 25% or 1/4 little h and little h. The other two blocks are going to have capital H and lowercase h, so 50% of them are going to be capital H, lowercase h. It tells you that if you have capital H, that's going to be heads, lowercase h is tails. And so according to this, you have a 75% chance of having a heads flipped and a 25% chance of flipping tails. Okay, so then you're going to start flipping your coins, and it tells you to do 100 coin tosses, and you need to tally up. Are they 2 heads, a heads and a tail, or are they 2 tails? And so you'll have like 3 columns, and you need to tally up. You should have 100 tosses, so you need to fill in like 1, 2, 3, 4, 5, and that's going to be your kind of matches your percents. Okay, So in your group data, your expected genotype, this is what we're going to get from this information. We expect 25%, 50%, and 25%. This is going to be where you put your total numbers, your final numbers. If you end up having like 23, 47, and 30, I can't do that kind of math, but just off the top of my head, then you're going to fill in what you actually got. How many flips over 100, and you're going to fill in there. And then this is going to require you guys to work somewhat quickly. The lab opens on Tuesday, April 14th, and you guys need to, we're going to have a deadline that we don't normally have, and that's that by Friday, April 17th, you guys need to share your data on Canvas, because if you don't share your data on Canvas, you don't have the class data. So that gives you Tuesday, Wednesday, Thursday, Friday, 4 days to get the data and share it on Canvas in the discussion board so you guys can do the class data. If you don't share your data, you're not going to get full credit. Okay? Then you're going to do the phenotypes. You're going to do the expected, which we said was 75, 25, what you actually observe, and then the class data. Okay, so that's going to be it for that page. For the check your understanding, you're going to answer all 5 questions, okay, on page number 147. All right, so then there's the fun part. Normally we do this as a lab, as an entire class, but we're going to do genetic traits. And going through, it's going to tell you to work with a lab partner, but because we're not together, what I want you to do is to work with your family, whoever you have, however many you have. It doesn't matter. I think you at least need probably 3. If you don't have someone, maybe you can FaceTime a cousin, FaceTime a sister or brother if you're not together, and go through these things together. But in order to have numbers that make sense, you probably need at least 3 for yourself. So you're going to work with your lab partner to determine the phenotypes for the traits and the procedures, and then they're based on 2 alleles, like everything we read. We're going to collect the class data on the board, and then you're going to share it. And so to go through them, it's going to tell you, like, look at this first one, the bent little finger. And so if you look at the image up here, it says, you can see it's asking you to look, does your pinky bend in? And so you can see mine's like a little bent, but it's not like that. So this would not be bent. Just because it's a little curved doesn't mean it's bent. So it tells you that if your last joint bends towards your ring finger, you have the dominant C. If it's straight, it's lowercase c. Okay, so then on here, because mine is mine is straight, mine would be little c, or mine would be straight, and then it would be little c on the genotype. The phenotype is what physical attribute it is. So you're going to put yours in here, and then you're going to write in how many people you're going to work with, and then you're going to have their genotypes and how many. Okay, so then let me go back. Okay, and then you're going to do tongue rolling, so you know whether or not you can go or not. If you can't, the widow's peak, so right up here. The free earlobes, you're looking at this little portion right here. Finger hair, whether or not you have that. Dimpled cheeks, eyebrow raising, ear wiggling. You know, like if you can sit there and wiggle them. I can't do it. My mouth moves, so I can't do that. Then it tells you your toe, long toe. You're looking at your second toe, whether or not you have curly hair, whether or not you have freckles. The only one you can't do is we can't do number 12, the PTC tasting, because you guys don't have the PTC paper at home. Okay, so okay, I hope that makes sense. If that doesn't make sense, then please ask me. You're not going to do the check your understanding questions after that table 8.18. It's on page 159. You're not going to do those check your understanding questions. Okay, now the last thing you can't do, because we don't have the blood typing, or you guys are at home, so you can't do the blood typing, is you can't do the blood groups on page 160 and 161 or 162. But I do want to explain blood types to y'all because it could show up as a competency to know. And so blood type is something that you inherit from your parents. Very similar. There's 3 blood types really. I mean, well, there's four, but it's A, B, O, and then you can have AB, which is a combination of A and B. Um, A and B, they can only donate blood to each other. So A can only donate to A, B can only donate to B. AB is considered to be the universal acceptor. They can take anybody's blood. They can take an AB blood, an A, B, or an O. Okay, so AB is universal acceptor. So it's great to have an AB blood type if you need a blood transfusion because you can take anybody's blood that's available. O, on the other hand, is more complicated. O type bloods can only have O blood. They can't take AB, A, or O. Only O type. But O type blood is the universal donor. So A and B and AB can all take O type blood. So it's, there's advantages. I'm a type A. I'm an A positive. So the positive and the negative, if you know your blood type, refer to an Rh protein that you inherit. I believe it's from your mom, but it only really becomes significant, one, when you need blood and when you're born. If you're a positive, then there's no complications when you're born. But if you're a negative, like your mom is a negative blood type, then there's a chance that some bad things can happen after childbirth. And so they usually will give you injections before and after childbirth to prevent those negative things from happening. So your body basically rejects, essentially. And so I'm not a biologist, so I'm not super familiar with it, but I do remember learning about it. And when I had my son, I got tested for that to make sure I didn't have it. And so you could be A positive, A negative, B positive, B negative. And those also have implications on who can donate. So it's kind of confusing a little bit about donations of blood, but you do need to know the different blood types, kind of the universal acceptor, universal donor, and understand that the genotype influences the phenotype, which is the actual blood type that you have. So my blood type is A, but my phenotype is A. My genotype could be AA or AO, depending on what my parents are. My mom's also a type A. I don't know my dad, so I don't know his blood type, but most likely he's probably an A. That's the only way I would have gotten an A blood type, or he's O type. He could have been AB as well, but he most likely was not a B type blood, because then I most likely would have had AB blood. So hopefully this helps you go through there. If you have questions about what you're supposed to be filling in, please ask on the discussion board. And then what I want you to do is for the commenting on the discussion board this week, I want you, one, to comment about your penny flipping. You have to do that by Friday, April 17th, midnight. And that's so that everybody can finish the data. But I want you to tell me one of the 11 traits that you're going to look at that surprised you, something you didn't know that was unique or different about yourself, and just share that. Okay, so each of those comments will be worth 2 points, and the lab itself is worth 6 points. So doing some or none obviously results in points being taken off, but doing all of it on time and completing it results, you should get a 10 out of 10. Okay. I hope this helps. I hope you're doing well, and please let me know if there's anything you need or anything I can help with regarding lab. We have one more lab after this, and then we'll be done with the lab portion of the class.